Okay, so now looking at capillary dynamics, just focusing on that portion of the vessels exam. So in this PowerPoint or this presentation, you'll see all the information you need to know to complete that portion of the exam. So first we'll look at how the pressures in general affect the movement of fluid across your capillary. So in order for your blood to get nutrients as well as oxygen to your tissues and remove waste from the cell, it's driven by two pressures. So at your arterial end, so on this end, it's driven by your net filtration pressure. On your venous end, it's driven by the net reabsorption pressure. So net filtration drives fluid out. Net reabsorption drives fluid into the capillaries. So there's two types of pressures that drive both of these, drive both of the net filtration as well as the net reabsorption pressures. So these two pressures are your hydrostatic pressure as well as your colloid osmotic pressure. So hydrostatic pressure is a pressure that a fluid exerts against a surface, and in our case, our surface is the capillary wall. For colloid osmotic pressure, so a colloid, if you remember from chapter 2, is a mixture of water and proteins. So when there's a difference in gradients of those proteins, so one side has higher amount of proteins than the other, it allows a draw from the other side of that membrane. So the colloid mixture inside your capillaries is the plasma. The colloid mixture on the outside of your capillaries are the fluids of the interstitial space. So because the capillary walls do not allow the proteins to travel across its membrane or through the gaps in between those endothelial cells that make up the capillaries, it causes osmosis to occur so that you can balance the water levels on each side of the semipermeable membrane, which in our case is a capillary wall. So we have our plasma, which will draw water into the capillaries, and our fluid of the interstitial space draws water out of the capillaries. So for your first pressure, your hydrostatic pressure, you have hydrostatic pressure of the blood as well as hydrostatic pressure of your interstitial fluid. So for your blood, the blood hydrostatic pressure is the force of the blood pushing against the capillaries, also just generically known as your blood pressure. So it's the pressure of the blood pushing out at the arterial end, and then by the time it reaches the venous end, it actually reduces. So in our little example case, the measurement is 30 millimeters of mercury out the capillaries, and then it drops to 10 millimeters of mercury of pressure measured at the venous end. For your interstitial fluid, the hydrostatic pressure is actually -3 typically in measurements. So this causes a suction, so pulling fluid out of the capillaries. So that's our 3 out here. So it actually helps to drive fluid out of your capillaries, even though it's a force of the fluid that's on the outside of the capillaries. So again, just a reminder, hydrostatic pressure of the interstitial fluid causes a suction to draw fluid out, and that doesn't change between your arterial and venous end. Moving on to your colloid osmotic pressure. So again, your blood has its own osmotic pressure as well as the colloid or the fluids of your interstitial space. So inside your blood, the colloid is plasma. So this draws into the cell a pressure of 28 millimeters of mercury. So this doesn't change between your arterial and venous end, since those proteins can't cross the membrane, so those levels don't change. For the interstitial fluid, it's a lot smaller effect, but still able to draw some water out of the capillaries, which again, since it doesn't cross that capillaries, those proteins are too big. So we have 8 out here as well as 8 out here, so no change between the arterial and venous ends. So how do we calculate those net pressures? So the two driving forces for delivering nutrients and picking up waste, you have your net filtration pressure at the arterial end, your net reabsorption pressure at the venous end. So both of those are a combination of your hydrostatic pressure as well as your osmotic pressures that we talked about just a little earlier. And again, the only pressure to change from the arterial end to the venous end is your blood hydrostatic pressure, or just blood pressure. So it lowers at the venous end. So to calculate your net filtration pressure, it's a combination of, so net filtration is on the arterial end, so a combination of your arterial end hydrostatic pressure as well as your arterial end colloid osmotic pressure. So for your arterial end hydrostatic pressure, so here, it's a combination of your blood pressure as well as the interstitial fluid pressure. So if your blood pressure is 30 out, and this is assisted by the suction from the interstitial fluid, so it also creates a pressure of 3 out. Combined, it makes a pressure of 33 out for the arterial end. So 30 out, 3 out equals 33 out. For your colloid osmotic pressure, blood COP 28N, so the draw of water into the capillaries from the plasma of the blood. And then for your interstitial fluid, draw of water out the capillaries into the extracellular space is only 8 out. So these counter each other. So with our arrows, we have our 28 in countered with a 8 out yields a 20 in, since the in has more pressure driving inward into your capillaries. So to get your net filtration pressure, it's a combination of both the hydrostatic pressure as well as your colloid osmotic pressure. So you have a 33 out combined with a 20 in. So those end up opposing each other. So you end up with 13 millimeters of mercury pressure that drives fluid out the capillary, since the 33 out is your larger number. Now moving to the venous end to calculate your net reabsorption pressure. So your net reabsorption pressure is your venous end hydrostatic pressure as well as your venous end colloid osmotic pressure. So combining them will allow you to get to your net filtration pressure. So first one, your venous hydrostatic pressure. Your blood pressure is dropped by this time. So we had 30 at your arterial end, and now it's down to 10. Your interstitial fluid pressure did not change, so still 3 out. So you combine 10 out with 3 out, and it equals 13 out. So 13 millimeters of mercury of pressure out, driving fluid out of your capillaries at the venous end due to hydrostatic pressure. For your colloid osmotic pressure, so again, these numbers didn't change, so your 28 in draw from your plasma in your blood, your 8 out draw from your interstitial fluid into the extracellular space. So again, these still oppose each other, so you end up with 20N, since 28N is the bigger number. And yeah, so since 28N, so there's more going in than out, so it's still 20N in that direction. So by combining them, you have your 13 out opposed by your 20N. So that leads you to have 7 millimeters of mercury of pressure driving fluid into the capillaries. So here's the example from your book showing you all the additions. So we have hydrostatic pressures up top, your colloid osmotic pressures down here at the bottom. For your net hydrostatic pressure, so that's dealing with your arterial N, which is your 33 out, and then 13 out for your venous N. For your colloid osmotic pressure, none of those pressures change, so it ends up with both of them being 20N. So net hydrostatic pressure of your arterial N with the net onconic pressure or net colloid osmotic pressure at the arterial N, so your 33 out opposed by your 20N leads to a 13 millimeters of mercury of pressure out the cell, so driving fluid out the cell. On your venous N, you have your net hydrostatic pressure of 13, so difference because of the difference in blood pressure, opposed by your 20N, so those opposing numbers ends up with a draw into the capillaries of a pressure of 7 millimeters of mercury into the cell, so driving those waste fluids as well as carbon dioxide back into the capillary so they can return to the heart through your venous system. Okay, so let's practice. So like how it would be on the test, sort of. The pictures won't be as pretty, but that doesn't matter. Okay, so first you have your initials for each pressure, then your direction